Good afternoon, everyone. The next session is Translating Emerging Technologies, Building Patient Trust and Real-World Impact. My name is Nigel Jamieson. I'm a clinical academic. I'm a surgeon in Glasgow. And I suppose I just want to give a bit of introduction why I'm standing here today at this session. I've had the privilege of sort of building out some of the spatial omic analysis capability in Glasgow over the last few years, and I'm proud to say the Living Laboratory and the Cygnus project has been really instrumental in enabling the development of that, and in terms of monetary support, staff support, and thanks to Ross and Ruth for really building that capability in Glasgow. And I'm really proud to say that that's enabled us to sort of maybe discuss the challenges that brings. These technologies hold a lot of promise. The options and the hope to predict outcome from patient biopsies, tailor treatment, predict prognosis, predict therapeutic response, but also raise critical questions regarding translation, trust and impact. And I think today we want to really see how we move beyond these elegant and high throughput technologies to the point where maybe we can discuss about the impact that may have from a patient perspective, and can we generate something that we can trust and clinicians can rely on. And today, we're going to on the panel today, we've got three people who have been really important, I think, in bringing this discussion to the fore. I'm going to Joanne Edwards on my left-hand side is going to introduce herself very briefly.
So I'm Joanne Edwards. I'm a professor of translational cancer pathology at the University of Glasgow. My research team's based at the Wilson Wall Cancer Research Centre in Glasgow Estate, but I'm also director of Glasgow Tissue Research Facility that's based here in the Queen Elizabeth, which aims to provide tissue solutions to scientists. And I'm also director of InSize, which is a programme of work that aims to use machine learning, AI and integrated omics to risk stratify patients that are at risk of developing colorectal polyps in the pre-cancer setting.
Thanks, Joanne. And Emma?
Hi, I'm Emma Parsons. I'm a researcher based at the University of Glasgow, but I also manage the Glasgow Colorectal Patient and Public Involvement and Engagement Group, which is a group comprised of patients with experience of bowel cancer, their family members, and charity representatives.
And Ron?
Hi, everyone. Great to be here. Flew in from San Francisco, so I'm a little bit tired. But I'm a physician scientist by training. Never practiced, joined a company called Recursion Pharma, which is one of the early technology AI-enabled drug discovery companies. Was there for 6 years, essentially from seed stage through IPO. And then about 3 years ago, founded Noetik, which is a company that really is built on the principles of using multimodal data to understand human cancer biology initially, hopefully, other disease biology ultimately. And really what we're doing is training, large foundation models based on vision transformers on these very, very deep stacks of data that we generate.
Thank you. Thanks, Ron. And I suppose this panel is going to be a little bit different because we're going to start with a patient video, which I think should go smoothly, and just to give some insight from a patient's perspective. And then following that, we're going to move on to a short talk by giving Ron the opportunity to articulate really what his company are doing with complex data sets.
Sorry, everybody.
Every 2 years, people in Scotland aged 50 to 74 are sent the at-home bowel cancer screening kit. I was one of them. Returning that kit started a 6-week roller coaster of being diagnosed with bowel cancer. I was immediately at appointments every week, leading up to a surgery, followed by 4 rounds of chemotherapy. But over 10 years later, I am still cancer-free. When I think about healthcare innovation, I think of new therapies or new ways of care, smarter, more compassionate ways to improve patient recovery. In my cancer journey, I benefited through innovations such as early diagnostic tools and a complex surgery. When I finished treatment, there was a sense of relief, but also a feeling of being left on my own. The transition from hospital to everyday life was difficult. I didn't always know who to contact about side effects or emotional worries, and appointments that felt very spaced out. Beyond the hospital, innovation could mean things like better follow-up systems or personalised diet and exercise support after treatment. Innovation is about ensuring that people like me have access to care that's proactive and centred around our everyday well-being. When the healthcare system listens to our experiences, it makes life after cancer easier and more dignified. Recovery isn't just about being cancer-free. It's about truly regaining your confidence. I would say to any healthcare researchers and companies, ask us how we're helping and what our day-to-day realities are to help design treatments, digital tools and support systems that truly fit around patients' lives. Remember that patients are people, not numbers or conditions, and that they have fears and feelings. Behind every study and behind every product is a person working to get their life back.
Thank you very much. No, I think that was very powerful. And as a clinician, it's amazing how the fears that we often have of asking patients to contribute to studies are immediately kind of disintegrate when you hear those words, and actually how much patients, I suppose, expect from us in terms of data scientists or clinical academics or scientists to be using to the maximal benefit of patients and either themselves or patient peer groups these opportunities for data and studying. And I suppose maybe my first question is maybe to Emma, and maybe just what do you think patients are most concerned about in terms of their data or samples being used, and how could we better build confidence?
I think patients, first of all, think about their data. They want to know what it's being used for, who has access to it, and why it's being used. And I think in a lot of cases, if you can explain clearly and concisely to them that it's an important research question that you have that will benefit patients in the future, then they are more comfortable with their data and their tissue being used. I think it's just being as honest and transparent with them as possible, and in explaining things to them, the research question in a way that they'll understand, trying to avoid the jargon and the acronyms that we all as researchers love to use, but that's not as accessible for them.
Just from my perspective, I think just where you've said, Emma, just reflecting on transparency, I think that's, whether that's any aspect of a patient's journey, from diagnosis to prognosis to treatment to complications, I think that's, and I think, it holds true in this aspect of it as well. And I think as long as patients are informed, in my experience, they often have, look favourably in this. It's when things come out of the blue almost, and I think that's where the challenges and the breakdown in communication and that information gap arises. But I think it's just, and I suppose that's I suppose from your perspective, Joanne, you've, and in terms of the governance aspect of all the work you've done over a number of years, how does consenting for tissue use, how has that evolved, and where do you think it should maybe progress to?
So when I first started using tissue in patient cohorts and data, it was pre the Greater Glasgow and Clyde Biorepository, and I had to write my own ethics and get site inspections, and but also I had the flexibility of going into pathology archives, pulling my own blocks, going and collecting data notes and constructing my own databases, and then anonymising that myself and holding the key myself. So, and that was all in an unconsented way. Now the biorepository has made that process much simpler for me, and they have their overarching ethics for use of surplus tissue, which makes things a lot easier. And through that framework, we can apply direct to them for the tissue that we require and the linked clinical data. But with that, we also are in a position that if we want to collect additional specimens, we can still do our own ethics and get research purpose specimens in that way.
Okay. And, Ron, maybe I'm going to ask one, this maybe question to you and just your thoughts, and then maybe have you come up and just kind of illustrate what you're doing at scale. But, how were your, thoughts as a clinician, ultimately trained as a clinician, and then, your practice is ultimately, huge and complex data sets. What are your, how do you as a company, build confidence in now, handling these data sets?
Kind of speaking to some of the previous points, we took a little bit of a different approach than a lot of folks building these data sets. So, we didn't, sort of try to build some business that's going to, bring us data in, as it were. What we did was we decided we were going to source, commercial tissue that's, completely unidentified, where we don't actually have very much clinical annotations. In many cases, these are FFPE that are 5 to 10 years old. So they're archival samples that we can essentially work with commercial biobanks to source. And so what that allows us to do is build a very large data set that we can train the models on without having to sort of establish a lot of collaborations to bring in small data sets. Obviously, you all have a lot more samples than most people we've spoken to. Now, in terms of your question was like, how do we think about using those?
Yeah.
And so really kind of our approach to building out these models is sort of the thesis is if we can train the models on enough data, then when it comes time to actually solve the clinical problem that we're trying to solve, where we are usually in a regime where we don't have enough data, then we can essentially use the model to interpret a smaller amount of samples or a smaller amount of information. So for example, one of the things that we are doing today is we've trained, large models on lung and colon cancer specimens, and we're interested in, understanding whether there are certain clinical programs that, would be of interest to us to license. And so we can work with the existing, data from those clinical trials, be they, biopsies or H&E. We can use our pre-trained model, and then we can essentially interpret, the small numbers of samples. Usually it's like, let's say 20 to 100 samples from these trials. And then we can use these models to predict responders or predict biomarkers that could inform us about response. And so really we're sort of leveraging this large data set that's unlabeled data to be able to essentially use and make use of and sort of enable a much smaller data set, which you would never be able to train a model on.
Okay. Have you got some slides to?
No,
that's fine. I will just, no, that's perfect. Maybe back to Joanne, I suppose, and what you've generated a lot of infrastructure over the last 20 so years, based on multiple and really aligning with what Ron's company has been utilising as tissue microarrays, which obviously presents challenges because you have multiple tissue samples being analysed simultaneously, archival tissue, this not consented specifically for this purpose. So some of these challenges we heard a little bit earlier from Charlie, come to the fore. And you really did that, ahead of time, and you were, generating these 20 years ago, and now these are becoming incredibly useful in these archival tissue samples. Maybe, how, how do you feel those cohorts are going to enable discovery for patients?
So really my aim as a translational scientist was always to be able to identify things in the lab that could be took into clinic and be clinical applicable. But to do that, we had to navigate challenges, not only from technical validation, but to regulatory approval. And how are we going to do that? So my thoughts were, I was based in the Department of Surgery. I was surrounded by access to really world-class human cohorts. Back in those days, every patient you had to do in an individual slide. Technically, that was very challenging for reproducibility. It was very expensive and it was very time-consuming. So how are we going to be able to leverage these large real-world clinical cohorts that were really enriched with granular data to enable us to identify these truly independent, targetable prognostic markers? And alongside that, I was really interested in deepening our understanding of molecular phenotypes that underpin direct pathology phenotypes that were known to be highly prognostic, but we didn't understand these, so we couldn't intervene in these. So I started working on tissue microarrays, which back then the main purpose was to get as many different patient specimens on the one slide at the same time. And it was literally sitting in a back room of the Royal Infirmary with a single-headed machine where I, myself, and then an intercalating student and a PhD student joined me, Nigel at the time, were coring blocks, one patient at a time, aligning it with an H&E, and it was an extremely lengthy process. But what that enabled us to do was have cohorts of thousands of patients that we could then put through pipelines such as IHC, and it really opened up the world of looking at large cohorts with granular clinical ability. Of course, now that's not really the main aim with tissue microarrays. Now, one of the key things that we do at Glasgow Tissue Research Facility is be agile to the needs of the new platforms that are coming through. So we still have all these large cohorts with all the granular clinical information, but we use these cohorts in a different way now. So we're really dynamic to be able to construct TMAs to the needs of the researcher to harness the highly complex new technologies that are coming through, like the spatial omics platforms that you're using, Nigel. So being able to adapt to the needs of the different platforms for the individual uses. And I think that's where we're really powerful.
Yeah, and I'm hoping that it's, that this what you describe is also scale, and it's about how we scale some of these things, because obviously for cancer patients there's often huge heterogeneity in a disease, and so that scale is necessary if we're going to study a disease. It's not enough to study a small portion of patients. We need to study a really large cohorts. And at the same time, how, I suppose, do we, you're describing a way of high throughput technology, and we can get a lot of data. And at Ron's end of the table, we have this, again, very high throughput investment, which enables really large study to move very quickly and really generate thousands of data points and even a small piece of tissue. Do we feel that data generation is moving faster than policy? And how do we keep up with that? Because even in the past, it was hard to generate a whole genome. It took a long time, took a lot of money. But now we can generate, whole transcriptomes on thousands of patients within a relatively short period of time. And how do we, from a patient perspective, how do we manage this sort of, speed of data generation, innovation, and maintaining trust? maybe to Ron?
We don't have a good way to maintain costs. We are raising venture financing.
So that's still a limiting factor. It's still a limiting
We are no, but I mean, so we started the company kind of with this. I'm not a pathologist. I didn't train in pathology. I've never worked with human tissues prior to starting the company. We basically said, okay, we know how to build machine learning data sets. So we want to solve this machine learning problem. And so we spent a lot of time, 6 months talking to people, talking to pathologists, understanding, some of the issues with spatial data, with histology, and learning about, some of the precedent in kind of the digital pathology space. So that was one piece. And then for the machine learning side, we knew we needed to be able to generate a certain type of data. So, images typically are much better for training models. So we felt that images would be a powerful data set, and we need to generate data at scale. And so from there, we sort of constructed, a solution to this problem that in many ways, might look a lot different, and in fact does look a lot different than what one might construct, maybe 10 years ago in the more of the digital pathology regime, where, a lot of people are kind of, more focused on whole slide images. And because what we needed to do was really capture as much data as we can from these very expensive assays. And so what we were trying to do is that, on one hand, reduce the cost of data generation by a multiple. So we were able to put many patients on a single slide to be able to generate, data that's, tens of thousands of dollars per slide in some cases. But also we needed to control for some of the things that you're going to run into with machine learning. So if you have an expensive assay, you're only going to run one slide, then you're going to have a batch effect and the model's just going to memorize. Essentially, there's no distinction between your patient and your assay run. So you need to have representations of patients on different slides. You need to have many different representations of patients. And so you sort of need to solve, both kind of the, biology, histology problems, and then also be careful with the machine learning problems. And in so doing, we sort of bring the cost down, almost an order of magnitude in terms of the assay cost. But end of the day, we still need to generate a lot of data. And it is quite expensive to generate the data. So, we, really kind of founded the company and said, well, the first thing we're going to do is generate, a large lung cancer data set, train the models, and, we'll see if it works. And it ended up, with paired multimodal data with not even, we were at, close to 500 patients and where we have H&E protein and spatial transcriptomics, and we were seeing, early signs that these models were learning the underlying biology. So we just kept pushing forward.
Okay, and maybe do you think from Emma, from your perspective, how this this data is increasingly complex. Maybe 20 years ago it was kind of singular. You may be explaining to the patient that in this analysis of your tissue sample, we would look at a gene or we would look at a protein, and that could be articulated in a straightforward way, and it was easily understood, and the impact was foreseen. How are we doing a good job now of explaining the complexities of what can be done with multi-omic analysis, with AI models, interrogating those with potentially an element of unknown algorithms running? How are we doing?
I think we could do it better. Speaking from the Glasgow Colorectal PPIE group, we showed them the Insights project that Joanne mentioned. We had a lovely cockpit and it looked lovely. It was very much patient-facing. And they said, but what's behind this? They really wanted to know. And it was trying to explain to them some really complicated features behind that. And actually, I sat down and thought, you know what, I think the patients in this group could use some training. We want them to give us advice, but actually they need the knowledge to be able to understand what we're asking them. And I knew I wasn't the person to give them that training. So I went away and found somebody who was an expert in digital pathology and AI, and I asked them to come along to the group. And that really gave them the knowledge and the expertise. They got a lot out of that session, and they could ask questions of an expert and really get somebody who's on the ground doing it, but that can also explain it in a way that was easy for them to understand. And giving them that expertise, I think, really allows them to interrogate what we're telling them, that we're showing them these new data sets, and they can actually then give us advice on that, and they can understand it a lot more. And so I think if we underestimate the patients, if we think, they won't understand it, so let's just give them the basics, I really think that's a mistake, and we should be educating them and giving them more knowledge so that they can help us with these more complicated things, and then we get a lot more out of them.
Okay, so would you agree instead, is going from a transition of them going from using patients or using patient samples to partnering with the patients and these things? And I suppose that, and I think, to overcome the barriers of, AI analysis, that we need to really bring them up to speed.
Definitely. Yeah, I think it's having those long-term relationships, keeping these patients in conversations with you. You've spent that time training them, you've asked them for advice on one project, can you get them involved in something else? We have them on as co-investigators on our grants now because we really value their expertise, and we get a lot out of them, but we're also putting a lot into them with the training and keeping them on board. And I think that's the best way of going forward with this, definitely.
Yeah, so it would, it's really a preparedness from our part, because I think it's not as, I think we all invest in clinical academics in terms of, having PPI groups and having people involved. But I feel that, what we're maybe asking them is what we asked them or involved them maybe 10 years ago. And the rapidity of what we're now doing is going so fast that we're struggling to keep up. And we're certainly not educating them in that way, in the same way, patients come into the clinic and they say, oh, I'm really sorry, I've Googled this. And I go, oh, that's really good. I'm glad you Googled it, because at least now we can have a kind of level of conversation. Whereas before we waste so much time trying to speed, bring them up when we're maybe consenting them, and that's not the right time to do it. So, from Joanne, I think we've, Emma mentioned the INSIZE thing. How do you feel that, I think INSIZE is a really important study that you did, in at a population level, essentially in Scotland and in the west of Scotland. Maybe is there some insights into kind of, how you've, kind of built that and, used, what the PPI work within that to help make impact?
I think from the PPI point of view, it was really important to take the patients on the journey from the beginning of INSIZE. So what INSIZE was aiming to do was to combine computational pathology, multi-omic data, and use an AI method to integrate it and identify a better way of risk stratification for patients with colorectal polyps. But it was a different approach. It wasn't going to offer the patients new treatment. It was going to actually identify patients that didn't require an invasive procedure. So it was actually going to identify patients that maybe didn't need a colonoscopy. So that it was really important to have patients on that journey from the beginning, because we didn't want patients to look at it or perceive it as being excluded or denying care, but to reflect a move towards an evidence-based decision to avoid unnecessary procedures, which would take away all the problems associated with not getting a colonoscopy if it wasn't required, and allow us to focus on the really high-risk patients that acquired these procedures. So to actually get the patient perspective on that from the very beginning was really, really useful. So it was not only considering the things that we would always consider, like clinical relevance, how would we integrate the workflow within the NHS, regulatory approval, but what did the actual patients think about this? Were they going to think, no, you're just denying us a test to save costs, or fully understand and be able to present it in a way that it really was to identify true low-risk patients that wouldn't require this invasive procedure, so that the focus could be on the true high-risk patients. So the PPI group that Emma set up for INSIZE, that's now developed into the colorectal PPI group, was really integral in that decision making and how we presented it, because it couldn't just be something that was ready for clinical adoption, that had clinical acceptability. It needed to be something that the patients would accept and feel it was making their polyp journey better and more acceptable. So I think that was really relevant.
And maybe just, following on from what that enabled and that sort of because I think that colorectal PBI group then facilitated on-treatment biopsy work as well, in a sense, with, Campbell Roxburgh's, Prime-RT study where, and standard of care study where biopsies were taken from, and this is an interesting kind of, opportunity to discuss, Ron, just in terms of some of the things we've layered on multi-omics into biopsy work, is that patients consented for on-treatment biopsies during their treatment of rectal cancer. So they had diagnosis and then they actually had serial colonoscopy performed every 4 to 6 weeks following immunotherapy, following radiotherapy. They got to witness their tumour reducing at the time of colonoscopy. And that helped them, sign up for this opportunity to have tissue collected serially. And that was obviously a lot of, back and forth with the patient PPI community to enable that. And I think if you'd said that 10 years ago, and how many patients would sign up for that, hardly any. Now we've got hundreds of patients with 3 or 4 time points. And then that was then introduced into an AstraZeneca trial. And so that data cohort is, now mature, trials almost published, and that will be analysed using this patient multi-omics approach. I think that's ultimately that building trust with the patient was the only way that could have been achieved. And you see it falters when a different clinician consents. And in a way, it's as fragile as that because, I think, you can, so I think these are the challenges though.
Well, what was really interesting about that was when we first closed that to the ethics committee, it got knocked back because they said patients will not consent to getting biopsies that aren't necessary for the treatment plan. And it was only by collecting the patient data and the patient point of view and representing that to the ethics committee to say, actually, patients really enjoy real time seeing how they respond to the therapy, and patients want that we're able to get it over the line with ethics. So it really shows how important it is to involve patients from the very beginning, because what a patient wants is often different to what we perceive a patient wants.
And following on from that, I don't know, back to Emma in terms of managing expectations. Do we oversell some of these emerging strategies, technologies, and so on? Because always again, it's this sort of expectation gap between what we're trying to offer to move things forward, but maybe at the same time, I always feel like sometimes uncomfortable with that. I don't know what your thoughts are.
I think sometimes, sorry to go back to the insights example, but we showed it to the patients and they said, this is brilliant, when can we sign up? And we had to explain to them, this is years down the line, this is going to be for patients in the future. But I think it is just being able to remind them that they're still having a massive impact on something that will affect patients, even though it is longer term down in the future. And I think, managing expectations is really important, especially with longer timelines like that.
I think it's also managing expectations with the researchers and for clinical implementation as well, because they don't always understand how difficult it is to get a new test into the clinic. For example, the Glasgow microenvironment score we first started working on in 2015, and we thought this is going to be easily implemented into clinic because it's based on an H&E slide that's already done routinely, and it's highly prognostic, and it can identify a group of patients that would respond to a certain type of chemotherapy. But because we spent 7 years gathering all the evidence and validating all different cohorts before we actually started the conversations with the NHS, how we'd implement that, we were quite naive and we were set back, and then we actually had to go back and do really simple things like use 4 different digital scanners to collect the data and show that our algorithms were going to work across 4 different scanning platforms. And this was required, and then we had to do a small prospective study. How's it going to link into the clinical pipeline? And where if we had actually worked with the clinical implementation at the very beginning, that would have all been done while we were doing all our external validation cohorts. So that study came from a sort of naive background, but these lessons learned from the very beginning, and SAIS has been working with the NHS for clinician acceptability and to work out how this will fit within the current NHS pipeline to be able to be adopted once it's ready for use. So it's also about training researchers to speak to the end users right at the beginning and not wait till they think their tool or their product or their device is ready, because often we get it wrong and we don't know what the actual doctors want.
Okay, so communication between all, not just patient education, but end user expectations is important. Just I think maybe just highlighting a strength. I think that what we're trying to achieve in Glasgow is this biopsy-based strategy across multiple disease types, whether it's cancer, whether it's in autoimmune and inflammation. Where the challenges of you, Ron, in terms of where you see emerging technologies, and we're focusing really on tissue-based technologies. Do you see that as being able to deliver in the future, or do you see that as very far into the future? I would hope that there was promise a decade ago that we would be doing that. I think that we weren't able to achieve what we wanted to a decade ago, but obviously paraffin embedded based technologies have really accelerated and have opening up a lot of opportunity. And we would, what are your thoughts?
Yeah, so we're a little bit different in that we're not going directly to clinicians. We're, focused on therapeutics, so identifying.
But that's an important space for the drug companies.
Yes, initially. So
neoadjuvant therapeutics.
And so really our burden of proof is how convinced are we in our results that we can move a program into the clinic. But where we've seen proof points already. So as we're a 3-year-old company, so relatively young. But the first evidence that models can learn biology that we saw was that we can take lung cancer patient samples, and then the models can, in a unsupervised way without any labels, discover the sets of patients that we know are responders to immune checkpoint inhibitors. And we know that because we have some labels for some responders. And they can actually do it. The models can, narrow down on those patients better than the clinical biomarker PD-L1. And all in a, unsupervised way or a self-supervised way without the label. So that was sort of the first thing that got us really excited. We're like, well, because you don't expect that to work. I mean, you don't expect any of this to work sometimes. And, that was a very shocking result. And we can actually look at, what the models were narrowing in on in those patients. And there was sort of known biology that we understand to be true of those patients that actually were the features that were the model was using to distinguish those. So that was sort of the first guidepost for us that okay, this type of approach is actually working. Models can learn biology without labels. And then as we've sort of push the frontier of that, we are, more recently, some of the work we've been doing in the last, this is really in the last 2 months, is we can take a biopsy H&E, we can infer the transcriptomics from the H&E alone, and this is a clinical sample, very small biopsy. We can infer the transcriptomics, and then we can essentially use that transcriptomics signal to run the biomarker analysis using the foundation model. And so this was something, this was an example that we kind of, like realistically as a company, we're like, okay, yeah, maybe one day we'll do this. And, we started trying it and it works. And, we're running a number, like, half a dozen programs right now where we are, where we have all this clinical data and we're actually, testing for biomarker hypotheses using this method. So, I think what's, big picture, this is sort of a machine learning regime that I think has been very surprising for, even folks in kind of the mainstream and, large language models and so on and so forth. And I think if you have the right data, and you can build the model architectures, and you can pay for the compute, which is very expensive. We're seeing results a lot more quickly. And this is my experience, having been at a previous version of a technology company, 10 years ago, and we weren't able to do these types of things, prior to these types of model architectures.
Okay. And is there any, just before we close out, I'll have one more question. Any questions from the floor of interest, either from a patient perspective or for Ron? Oh, sorry. Technology not working.
Okay,
Oh,
working now.
You're on.
Thank
you're
you.
online.
Always be nice to the technical team. Interesting discussion. I'm interested to hear, maybe from Emma, but from anyone, of an example where the patient's told you not to do something and you didn't do it. We generally focus on the positive. And if you guys don't do that in cancer, I'll share plenty of examples of arthritis patients telling me not to do things, and we've long since learned to pay attention.
I don't know that I have any examples of that, actually. Joanne, do you?
They sometimes say to us about the big complicated data sets, are they actually ever going to work, and can we make it simpler? So it's not that they told us not to use the complex data sets, but we listened to them saying, make it simpler, and we took all that data as discovery data and we tried to unpick it and make it as simple as we possibly could. And actually, in reflection, that was totally the right thing to do, because it's going to be much easier to implement simpler things than it is complicated full transcriptome spatial cosmics, for example. The NHS is not ready for that type of data yet.
Absolutely not. No. Do you have an example? You have a case.
Yeah, when I wrote the renewal for our UK Arthritis Centre grant, so that was a huge grant, and sent it to our patient partners. We have 14 patient partners in our group, and they phoned me about a week later to say, this is terrible, Ian. You're trying to prevent rheumatoid arthritis. What about the hundreds of thousands of people in the UK already have the disease? So you need to write this again. And we did. We went back to a blank sheet of paper and we rewrote it in the following month. I think trust is built when you not only listen to your patients, but you change your plan on the basis of what we say. Otherwise, all we're doing is having them agree with us to make us feel better.
Emma, also, the patients pointed out to us that one of our PPI groups, they didn't like the language we used, and we had to change the phrasing that we used, and it was you that implemented that.
Yeah, you have quite a lot of opinions about the language we use, and sometimes you can change it, and sometimes you have to say, no, that's what the ethics committee want us to say. And we just have to pick our battles and try and be try and do as much of what they ask, but we can't always do everything.
Any other questions? And maybe just last point, what barriers do you think we still need to overcome then? And what do you think to make progress in terms of, maximising patients' trust and making impact from this emerging technologies?
I think a lot of researchers aren't getting the patients involved early enough. We have people come to us with quite well-established projects that they want to speak to the patients about, and the patients say, why have you not come to speak to us 6 months, a year ago? We would have had opinions about the whole design of this. You're showing us the results. So I think it's getting people speaking to each other early enough, and really recognising that what the patients say can be really, really important, even at a very early stage of the project.
Okay, so thank you very much for the panel for just discussing these points, and thanks for Ron for coming over from San Francisco, and hopefully there's an opportunity to engage with this panel just over the next few hours before the end of the day. So thank you everyone. Just join together and congratulate them.